Thank you, Claudia. Thank you so much for giving me the opportunity to present here. And thanks, Marty, for setting the stage here. So it makes my job easier now. So I don't have to delve too much into EPR imaging. I'll focus more on other application. So oxygen is a fundamental physiological parameter. That means we'll definitely have more applications. It's useful everywhere. So what I want to focus is, oops, I can change. I want to focus on another application is type 1 diabetes. I'll set the stage why we need the instrument, reliable and robust instrumentation. So I'll little bit give you a background. So type 1 diabetes is an autoimmune disease where body's immune system destroys insulin generating beta cell. So what happens in normal healthy people, we have this beta cells which secretes insulin in response of glucose, and that's how and that's needed by cells, and that's how it functions. However, in type 1 diabetes patient, this insulin generating beta cells are destroyed by body's immune system. And that means these patients cannot generate insulin and they need constant injection of insulin. One way to resolve this issue is by using bioartificial pancreas or beta cell encapsulation devices where we can implant these insulin generating beta cells in an immune protective environment and then implant it in patient. And idea is that then patient don't need insulin injection and they can maintain the normal glucose level. However, there are two factors here is important. First is that islet functionality depends on oxygen consumption rate. There is a lot of work by Dr. Papa's group at University of Arizona showing that oxygen consumption rate is really, really important. Another important point about these cells are that these are extremely, extremely sensitive cells on oxygenation. And most of those islet cells are lost within 24 hours of implantation due to hypoxia damage. They also get damaged by hyperoxia, but most of the loss is due to the hypoxia. So the number of groups are working on addressing the oxygen need of islets. And the common strategy is that you can increase surface to volume ratio so that you have cells have more access to oxygen and nutrients. Some groups and even companies working in oxygen generating in situ. So you can generate oxygen, and some groups working on external delivery. Some of these devices are in clinical trials, and some of these devices like Therasite were in clinical trial, but they failed. And so and here are these devices which we have looked in our using Jiva 25 or in our oxygen measurement course. So there is a strong need of oxygen measurement in this field. So I already covered that it's a challenge in cell therapy, not only in cell therapy, in tissue engineering, tissue therapies and organ replacement, everywhere we need to know the oxygen. And it's because we need to major issue is the quantifying oxygen. So we cannot mitigate hypoxia if we don't know what the oxygen level is. So with that focus, Marty has done a great job, so I will not go too much into detail. So this is EPROI. I'm focusing only on oxygenation, not other application of EPRI. Here I'm showing some maps of EPROI applied to the tissue engineering or the cell therapy products, showing how oxygenation changes. I'll go more deep into this later. Again, I won't go much into detail. We are measuring electrons, and measurement target is electron spins. Key difference between MRI and EPRI is magnetic moment of proton. And because of the high magnetic moment of proton, we need much smaller magnetic field. And Marty has covered that we also have very small relaxation time here. So strategies of image acquisitions are different. And since we don't have free electron, we have to have an exogenous probes. Uh, Trital we have covered in this. So trital oxo 6371, they are sensitive to oxygen. They dissolve in fluid. So when we are doing in vivo experiment, we can inject systemically, or if we're doing in vitro experiment, we just add trital to the sample of interest. And it's stable, water soluble, it does not enter into cells. Particulates solid probes here. Again, we are measuring oxygen concentration in the material probes. And since because of safety reason, we encapsulate into this bioinert oxygen permeable polymer. And I'm happy to report that we are synthesizing OXO 6371, and we recently also have first synthesis of LIPC available in the lab. This is non-toxic to cells. In vivo toxicity has been reported a long time ago, and it has been there are thousands of animals have passed through this trial. But we started doing, since we're doing cell culture tissue engineering work, we also measured in vitro sensitivity with cell, and it's pretty non-toxic to cell concentration. Here we did perform measurement up to 8 millimolar, but typically we use half to 1 millimolar in vitro measurement. So we know that both R1 and R2 are linearly proportional to PO2, and similarly at low field at 25 millitesla also we have low concentration dependence for R1 compared to R2. EPROI is precise. We did perform measurement with Oxylite Pro. This is the single point Pro, and for the various value of between 0 to 21%. And we can see that especially when we go to the higher oxygen values, we have much higher error with Oxylite Pro compared to EPRI. And I believe it's because these probes are not calibrated for various settings or various biomaterials. So we did perform measurement using DI water, gelatin, and some Min6 cells in medium. A little bit about choice of magnetic field. So signal to noise ratio grows with imager frequency. However, power absorption also grows with imager frequency. So we have to make a balance between the getting the maximum SNR with uh, maximum penetration depth. So if we take 25 millitesla, which is about 700 megahertz, that's good enough for mice, rats, etc. However, if we want to measure humans, we have to go to the lower field, and here we are developing a 9 millitesla human size instrument, which potentially, hopefully, will go to the humans. We can go higher field to gain SNR, but then we lose the penetration depth. So that's why this 700 megahertz is a sweet spot for small animal imaging. With that, I want to introduce our world's first dedicated oxygen imager. So right from the beginning, we were focused on having optimizing everything for oxygen imaging. And my goal is, or our goal is to bring this technology not only to the labs which are comfortable with MR or EPR, but to the biologists, to the cancer people, or to tissue engineering people. So we were optimizing everything so it can be a single click button operation. We are not there. We are there, like we are slowly making progress to making it as simple as possible. It's a small footprint. You can plug it in any corner. No cooling cryogen required because we are using a permanent magnet. We have registration tools available currently on MATLAB, but slowly we will transition it to the main software of the instrument. And one advantage I would say, Marty mentioned about the easy access to the magnet and animal, but we also have vertical access because many time biomaterial measurement needs vertical access. Simply also calibration needs vertical access. So that's why instrument is accessible both horizontally as well as vertically. And I just wanted to mention that Jiva word is has its root in Sanskrit, and it means life or being alive. So for oxygen imager, this is an apt name. We need oxygen for surviving. Uh, here I want to make a shout out to Martina's lab. We'll have the first deployment in Europe at Martina's lab, hopefully in few weeks from now. We are preparing the instrument to be shipped. Maybe in couple weeks, it will be out of our facility. So very, very excited for this new collaboration. And it's a shout out for Claudia also to create a node for oxygen imaging in Poland. So few more key critical points here, or key advantage, what I will say, or key for our instrument. So this resonator unit is swappable. We want to have the smallest resonator needed for the for the sample size. So if we have 10 millimeter sample, we don't want to use 32 millimeter resonator to reduce the losses. So these units are swappable. We can replace it, change it, and have the vertical axis or horizontal axis. Here is an example of a vertical axis resonator, which we use for calibration purpose because we calibrate every sample it passes through our lab. And this is the showing the horizontal resonator with a mouse inside. And 32 millimeters, so mouse doesn't get squeezed, and we have the full body imaging. We also developed the smart animal life support system, so we can keep animals at 37C throughout the experiment, however long it takes. It takes about 1 to 2 minutes to reach to target temperature and maintains it there using the active feedback loop. We also monitor animal breathing rate. And the way we do in our lab, we have one such system in animal prep room, and another system is obviously connected to the instrument. So you start monitoring animal vitals right while you are preparing, and then you unplug from one system and bring it to the machine, so it makes it very smooth operation. We have an operational video. I can put it in the chat after this talk, so if you want to see how to bring animal in and out of the system. We have developed the animal beds with fiducials, phantoms, calibration phantoms, because I believe in quantitative measurement. If we are measuring oxygen, I want to know that this is what it is. So this is the PO2 phantom example. This is example of concentration phantom to check the performance of instrument. Little bit on software. This is the software front end. You see the button here, tune, cavity profile. So you just press button and get the images you want. About spatial and temporal resolution. So this is actually image from SPI. That's why it's much more higher resolution. But using iris, we can get about 250 micron in vitro. In vivo resolution is about 1 millimeter. Oxygen resolution 1 torr at hypoxic condition and 3 to 5 torr in hyperoxic condition. And imaging time could be as short as 1 minute and as long as hour, depends on the resolution you need. And here you can appreciate the high resolution of SPI. So this is a device loaded here, and you can see the device in the middle. This is the bubbling line, and very beautiful. And this is the PO2 map because device was loaded with cells, so you can see that cells are consuming oxygen. So we can get, since it's a imager, but it's also a spectrometer, we can get average oxygen as well as oxygen maps. Uh, try as I added this slide just in the middle of Marty's talk. So tritral injection, we can do IV versus IP, and we have seen that IP provides higher SNR, and as long as we keep the breathing rate constant, we can have the average mouse VO2 constant. And this is also I added in response to Marty's comment about the variation. Some of that variation could be because of the cyclic nature of oxygen in the body. So maybe tumor variation, what you are seeing, it could be the cyclic variation in the body. Here, just showing the healthy few voxel of the healthy animal, showing how it's changing, where the average mouse PO2 is constant. Since we deal with a lot of biomaterials, we perform calibration and add all this calibration files into the instrument. So gelatin, agar, alginate, these are first few of them, but we have added many more and at physiological range. So all of these calibration is added into the file, so make it easier for people to use the instrument as well as the right calibration files. So I'll give some example. I don't know how I'm doing time wise, if I have time to show some example. So one example, we can use it for the biomaterial quality control. So biomaterials have different oxygen diffusion rate. And here is an example of gelatin. Just a simple example, we deoxygenated gelatin, freshly prepared or overnight refrigerated gelatin, and you can see the different oxygen diffusion profile. And this is the map showing that when we have the overnight refrigerated gelatin, even after 16 hours, it's not fully oxygenated. we do a lot of collaboration as part of our oxygen measurement core, and I will just show a few example of how it's used in the biomaterials. This is a collaboration with Matthew Matthews Group, and they have the hypoxia-inducing biomaterial for various applications in tissue engineering. So we perform measurement for that for them and confirm how it's changing, and this was published recently. This is a collaboration with a company called Ronawak Inc. and they have 3D cell culture system. They call it BioBlock, and we perform measurement for them. And this is an ongoing collaboration. We plan to do the cell loaded. This is acellular measurement showing beautiful how oxygen is in this 3D printed biomaterial, and we plan to load with cells and to continue the measurement in coming weeks and months. This was collaboration with University of Connecticut group of Dr. Nukawa Rapu, and they are making osteochondral tissue based on PLGA with various porosity, and we perform measurement for them showing how partial oxygen pressure changes as a function of porosity in the biomaterial. Now returning back to the type 1 diabetes, I will just show a couple of example here. These are the devices from our collaborator. So I'll just show briefly what we did, what kind of measurement we perform using Jiva on these devices. So first example is from the group of Minglin Ma at Cornell University, and they've developed a device inspired by insect, which has the high oxygen diffusivity. And you couldn't get this measurement or these numbers from any other method if you can see here. So this is a control device. We deoxygenate it first and then we let it oxygenate. And you can see that in this control device where nothing is happening, oxygen is diffusing from top to bottom. However, when we have this active device, you can see how oxygen is diffusing in the bottle here and it's getting much faster oxygenated. And we published this work last year in Nature Communications. Another example from the same group is the IBED device, inverse breathing encapsulation device, and it's a very clever device. So it relies on cellular respiration. So cells produce carbon dioxide, and this is lithium peroxide based device which reacts with carbon dioxide and produces oxygenation. And we perform measurement for them, confirming that active device does produce oxygenation, and this was published also last year in Science Advances. This is another device, Therasite. It's considered the industry or field standard in type 1 diabetes, and we did a measurement using various cell density. And manufacturers say that you can load up to 10 million cells, and when we load with 10 million beta TC6 cells, device quickly became hypoxic, and we lose the cells when we do this kind of experiment because cells do not survive this kind of hypoxia for a long time. And last example I want to give is the oxysite from Sherry Stabler's group at University of Florida. So they have oxygen generating device. There is a disk in the middle. Here you can see in this picture, and this is generating oxygen. And we first confirm with the in vitro test. This is at week 2. This is week 4, showing that week 2 it still have about 100 torr in this device, and week 4 about 50 torr. So we did in vitro measurement. This is cell-loaded oxysite construct. You can see that control construct when you have the cells, it's fully hypoxic construct. But when we have the active construct, there is oxygen generating here, and there is surrounding cells getting oxygen here. And this is the media PO2 and various statistics of various area. I'll just show a small example of in vivo measurement of this construct loaded into the diabetic mice model, STZ. So this is 1 day after the implantation. We loaded the device with 500 freshly isolated rat islets, and you can see this is the device. Here it's a hypoxic device. Cells are kind of hypoxic. They do not get oxygen. this is a control construct, it's not oxygen generating. And then but in second animal, this device is not hypoxic. Most probably cells were dead by day one when we measured them. So this shows importance of measuring. And if we can get this information 1 or 2 days after implantation, we don't have to complete the study or go far up to 4 or 6 week or 8 week and when the device fails. So this is a very nice example showing why it's important to no oxygenation for any cell encapsulation device or tissue graft right at the time of implantation, especially first few days are very, very important. So in summary, I think both Marty and we are convinced that it's an excellent technique. We have developed Jiva 25, very robust instrument, and a big shout out to Boris for all his kind of effort last few years to making sure that instrumentation is top notch. Measurement range is about 0 to 162. We cannot go beyond too much because relaxation days uh, values gets too short. Relaxation times is too short to uh, get this good SNR using pulse method. Currently, we showed only cancer, type 1 diabetes, tissue engineering, regenerative medicine, but I think world is open there. There is a lot more application we'll find as we move forward. And I want to thank we have support of lots and lots of people over the years. So, but I want to thank key collaborator. Marty has been doing tremendous job at MD Anderson. Viresh has been instrumental helping us making Trital. We using SPI technology from Murali Krishna's lab, type 1 diabetes colleague, Minglin Ma, Sherry Stabler, Dr. Papas, Clericos Papas, and Carl Simon from NIST. He has been very instrumental. We are developing a non-viability cell assay using oxygen imaging. I hope I'll be ready to show those results next year or so. And this is our excellent team and funding agency, obviously, NIH, NSF, and JDRF. And with that, I'm happy to take any questions you have.